Hello everyone, I'm speaker Cai Shaoxiang, and today I'm going to introduce this large field of view microscope image for nonlinear microscopy. And here is my outline, and I will paper review and motivation, design, method, results, conclusion, and final the future work and reference. And first, we are talking about the field of view. And microscopic field of view is the maximum area visible when looking through the microscope, usually as a diameter measurement. You can see the figure here. And maximizing field of view is desirable for many applications because the increased resource in more data collected, which gives a better statistical measurements for detecting the subtle effects and also decrease time needed at the microscopy. And here is the field of view formula. And my paper review is about ultra-large field of view two-photon microscopy. And it presents a two-photon microscopy that images full extent of the neural cortex with an object limit spatial resolution across 8 mm by 10 mm field. And the lateral resolution is 1 micrometer, and the maximum scan speed is 5 millimeters per millisecond. And the scan pathway employs the large diameter compound lenses to minimize the aberrations. By recording resting states vessel motion across both hemispheres of the mirror brain and image histological sections without the need to stitch. And the motivation, the mammalian brain is coordinated neural activities that spans multiple areas within the hemisphere and the functionally related area across the both hemispheres. The relationship between the blood oxygen, vascular dynamics, and the neural activity is a topic of a concerted study, and this implies the need for a microscope that can resolve micrometer scope changes in the vessel diameter across all of the neural cortex and is further compatible with the electrical physiological measurement. And in this page, I will introduce what is TPLSM. TPLSM, it means two-photon laser scanning microscopy. And in vivo, two-photon laser scanning microscopy is the ideal platform for optical sectioning through scanning tissue to resolve individual vessels and cells. And the ob objective that produce large field of view through high resolution require both large input beams to attain their full spatial resolution and large angular deflections of the input beam to make full use of available field of view. And this combination will result in aberration within the scan path that substantially decrease the resolution that may be obtained with the microscope. And here we are talking about design the ultra-large field of view TPLSM. And the illustration of a scan-induced aberration through the spherical single-lead lens in a configuration that corresponds to a scan length, and the on-axis scan beam coverage at the partial focal plane, but the off-axis scan beam coverage closer to the lens with separated horizontal and the vertical focal. And the focal plane intensity maps explicitly shows the resulting astigmatism. And this paper presents a design to mitigate aberration for brain-wide image of neural cortex with two-photon laser scanning microscopy. And you can see the figure here, utilizing a unit gain telescope to relay the image of the X-scan mirror onto the Y-scan mirror. We can see the X-scan mirror and Y-scan mirror here, and the separation of the mirror permits a separately compensate for scan-induced aberration along X and Y axes, and then a match length pairs is inserted, the X compensation inserted and Y scan compensation insert to the scan system to compensate for the net on the excess spherical aberration, and below is the parameter of the design. In this slide, a critical aspect of two-photon microscopy is the efficient capture and detecting of the emitted fluorescence. Highly scattering samples, such as brain, lead to emitted fluorescence that spans the full range of exact angles from the back aperture of the objective, or 6.4 degrees for our systems. And we can see the figure A. We use a compound collimating lens to capture all of the light returned through the objective, followed by compound collection lens, with allowance for bandpass filter to coverage the light to the photocathode of the photomultiplier tube. And you can see the picture B. The efficiency of these collection systems was considered up to the angle of approximately 5 degrees and dropped to a factor of 0.85 for the extreme angles of 6.5 degrees. And the beam characteristic in the figure A, it is the experimental verification of the beam collimatation. The rotation of the interference fringe at large scan angles found from the slope of the fringe as the yellow line here is consistent with the residual beam divergence of less than 1 million radian. And the B, the data shows in figure B is about the envelope evaluated through the four optical paths of the microscope at the focus of the object, and it calculates the rate fit to the envelope of the experiment blue. The data is consistent with the initial pulse width of 110 femtoseconds that is chirped to the final width is 400 femtoseconds. An application is two-photon image of a fixed and clear coronal section from a mouse that is spread enhanced screen fluorescent protein in a spare subset of cortical and hippocampal neurons. It obtains cellular resolution on and off axis such as the individual neurons are well isolated, both laterally and axially. And the figure C here, the Z projection are across 60 micrometers of a tissue at different depths into the tissue section, and the Y projections is across 50 micrometers of tissue. And the highlight of the hippocampal regions, yellow box in figure A, and the highlight of the hippocampal cells from figure A. And here is my conclusion. First, the ultra-large field of view two-photon microscopy maintains sufficient lateral resolution to resolve cells and vessels across 10 millimeters field of view. And second, the primary strength is the ability to image brain activity in tissue regions separated by distance. Third, and this shows that the whole tissue may be imaged without the need to stage smaller field of view together, and the circumvents the need to correct the differential distortions at the edge of a tailed fields. For the future, first,
A weakness of this microscopy is the reduced spatial resolution compared to higher numerical aperture objectives, which objectives is typically used for two-photon laser scanning microscope. And second, it takes advantage of future improvements in the designs of the low magnification, higher numerical aperture objectives. And the third, the principle of the designs yields a large diameter scan beam that is free of astigmatism across a wide range of scan angle and suitable for any infinity conjugated objective. And here is my reference. Thanks for your attention.